So I wanted to start really by providing a little bit of historical background to set the stage for the rest of our speakers, and then just give you an update where we are relative to incorporating this information into national cattle evaluation. So just to start off, right out of the box, right? 50K, HD, GGP, they don't come telling us how much or how well they predict a particular trait in a particular breed. We have to train those assays, essentially asking the question, how much of the variation can we describe using a given assay for a particular trait in a particular population? And generally speaking, we've done this using purebred training data. So training essentially on EPDs, or really deregressed EPDs, on a set of animals that hopefully are relatively high accuracy, at least moderately accurate. They're also genotyped. And so because of that, we've been pretty effective in describing a fair proportion of the genetic variation for traits for which we have dense phenotypes recorded, those traits that are already in existence in our national cattle evaluation. However, there are some phenotypes, and some of those will be discussed here today. Feed efficiency, Allison, BRD, and the list could go down in terms of novel phenotypes which aren't routinely recorded. As a consequence, we don't currently have EPDs for them, and a lot of those phenotypes are actually recorded on composite or crossbred animals, in which case the training often occurs on an individual animal's phenotype. Now, if you had a choice of a group of 1,000 animals for training that had high accuracy EPDs, or 1,000 animals that had individual phenotypes, Jerry, which do you pick? EPDs, because an EPD on a high accuracy animal is more than just one phenotype, isn't it? A lot of phenotypic information came into that EPD, and because of that, it's a little bit more powerful. So that the process of training, or discovery as we might call it, has received an awful lot of attention, not just the work by a lot of breeders and breed associations in this room, but the scientific community as well in developing methodology to make the best use of it. Steve Cashman back here. Steve, it's 2013 still, so we've still got a chance before we got to change that date to 2014. That's an inside joke for those of us that are on this paper. But we know that these tools, there's breed specificity, okay? Here what we have is, these are 50K derived molecular breeding values for the trait of weaning weight. And if our training population is purebred Angus cattle, and we predict in purebred Angus cattle, we do substantially better than if we try to transfer that information across breeds that were not contained in our training data, even for a closely related breed like Red Angus. So a huge degree of breed specificity. And I think most people in this room hopefully say, duh, right? 5 years ago, most people in this room didn't say duh, right? There was the hope, at least, that that was not the case. Now, we do know, though, that if we pool breeds in our training data set, we can actually do okay and develop genomic predictors that are a bit more robust across breeds. Here what we have is pooling purebred animals in training data for ribeye area, and those breeds used in training were Angus, Simmental, Hereford, and Limousin. And you can see that the genetic correlations are fairly robust across breeds that were actually contained in training. But as we go to a breed that was not contained in our training data, like Gelbvieh, we see a substantial reduction in the genetic correlation. So again, showing the sensitivity of these genomic predictors to the relationship with the training data, okay? If I try to predict the weather on the East Coast based on the weather here in Clay Center, how am I going to do? Not as well as if I'm trying to predict the weather in Friend, Nebraska. Who knows where Friend, Nebraska is? Yeah, darn right. Home of the world's smallest police station. We're renovating, by the way. We had to renovate because the Van Edenham stayed with me, and now they need a bigger police station. But at any rate, if we're predicting something close by, we do a better job. Okay? So we have to have breeds contained in training if we're going to try to predict in those breeds. So there's 4 general approaches, and I've gone through this before, relative to methodology of incorporating genomic information into national cattle evaluation. And our beef breed associations are really focusing on these first few, depending on who they are, fitting that information as a correlative trait, which perhaps at a 30,000-foot view seems the same way that we treat other indicator traits, although MBVs don't behave quite the same as other phenotypic indicator traits that we might think of in terms of ultrasound. But in general, fitting it as a correlated trait blending, which is a novel way of saying indexing. We're indexing a traditional MBV or a traditional EPD and the molecular indicator. American Hereford Association actually uses this, treating it as an external piece of information. So a breed like Simmental, which through their multi-breed evaluation has brought in data from other breeds, perhaps treating a molecular breeding value the same way, or what some other species do, but the beef industry to date has chosen not to, is actually fitting a genomic relationship matrix. And at one time, I had under here the caveat that you have to have access to the genotypes to use this approach. At one time, that was the crux. That's not the crux anymore. We actually have access to the genotypes. And I think that shows an evolution in terms of what we've been able to do in the beef industry. And none of these are perfect, admittedly. We could go around the room and everyone could poke a hole in any one of these approaches, all right? But the fact is, as I go through this, you'll see we've made substantial evolution in terms of methodology, in terms of building training data sets, and to your guys' credit, in terms of deployment of this information as well. I remember a few years ago, I gave a slide similar to this. At least the Angus breeders in the room recognize this animal. Where's Bill Rischel, right? People know the picture of him. I've made him maybe just a little bit different because I've changed the picture some, but that's Midian Focus, very high accuracy AI sire, all right? Extremely high accuracy. I asked the question a few years ago, if we also genotyped him with the 50K assay, and I provided you MBVs on this bull as well, would you make use of those MBVs for traits where he already has EPDs? And most of the room said yes. I would want that additional information. I think we now know, and based off those survey results, that molecular information tells us nothing new about this bull. He's high accuracy already. That adds that extra molecular information for traits where we already have EPDs, adds nothing to our knowledge of that animal. And that's depicted in this bar chart here, where the dark gray bars are the accuracy of an EPD before we include genomic information. The light gray bar is the gain in accuracy after we do so. This assumes a test that explains about 40% of the genetic variation. That's synonymous with a genetic correlation of about 0.6. You square the genetic correlation, that gives you the proportion of variation explained. So for high accuracy sires, this is adding nothing to our predictive ability. But for low accuracy sires, and even some that have already had some progeny, it does add some additional information. And I think what's really exciting about this, and we'll learn more about it at the end of the day, is for a trait where we have no other information, it adds a substantial amount. For a trait where we have no other information, some of those novel traits I mentioned early on, it adds a substantial amount of information. And I think that's the exciting part about it. You'll see on the program we're going to talk about BRD and feed efficiency. So why in the world would the scientific community not start with those traits? Why don't we have genetic or genomic predictors for those traits right away? Well, it goes back to that whole training process. We didn't have the phenotypes. We didn't have the phenotypes to develop the genomic predictors, so we started with the traits where we did have dense phenotypes, weights, carcass merit. So the current status, I stole this, adapted, I think is the correct term, from Dorian Garrick, right? Which essentially shows the breed, the number of 50K samples that he currently has in his repository, and whether or not they have deployed or implemented this in their national cattle evaluation to date. I think I'm in error here. I think Limousine should fall in the nearing category. But folks, that's exciting. 2009, right? Angus really was first out of the gate. And look at where we are now with a variety of breeds. And I think that's where I give the beef industry credit. We've taken a technology and we've put it to work, and you can see several others are working towards that end. And that has been enabled by partnerships. Okay, I think that's the exciting thing. True partnership between the industry and academic communities. The 2000 Bull Project here at USMARC, genotyping a little over 2,000 industry animals. The Weight Trait Project, which was really the genesis of this meeting to date, 50K genotypes over about 1,200 animals, and those fell right into those respective breeds training data. And then you see numerous federal grant-supported projects, whole genome-enabled selection grant, an integration grant deploying to the industry, trait-specific grants here, BIGS, grad students, if you haven't heard of yet, you will, GenCell, right, methodology for doing discovery. All of that enabled by some of these partnerships, which I think is extremely exciting, helping in deployment as well as discovery. And again, here I list the breeds with genomic EPDs. On the previous table, I did not mention Brahman, but focusing on the trait of tenderness, a trait that they have a keen interest in changing, actually producing genomically enabled EPDs for that. And the two primary players, if you will, in the arena of providing the service of getting these MBVs. So we have seen considerable advancements in terms of statistical methodology related to discovery and also implementation. We'll see from the next speaker considerable evolution in terms of assays, all right, the SNP panels that we have. And I've heard it referred to as cell phones before, right? A cell phone doesn't stay current very long, does it? It's continually evolving, and that's the way we need to view this technology as well. There's a lot of effort going into not only building discovery populations for the routinely recorded traits, but also those novel traits as well. Large worldwide sequencing effort going on. So doing whole genome sequencing, getting millions of SNP reads. Low-cost genotyping, right? I think for a long time, and perhaps in some people's minds still, that's the primary crux of technology adoption. But like all technology, it's getting cheaper. And one could argue that genomic technology, particularly as we look at the price of sequencing, is getting cheaper than we would normally expect. Okay? The price is decaying at a faster rate. Industry adoption. Actually, sometimes I'm labeled as being pessimistic, and I'm going to give you another word that I've been labeled here in another slide. It's child safe. But I think the industry has done a good job at gravitating towards this, building resource populations, actually adopting the technology. And the nice thing is phenotypic data collection has not been sacrificed in the process. We've done a good job, I'd like to think, but certainly the industry has done a good job at realizing that genomics does not replace phenotypic data collection, and it's just as critical as ever. Next steps, we need to do more work relative to novel traits, continuing to build those resource populations and also doing discovery there. Transfer across populations, okay? Composite animals across breeds. There's a lot of methodology work that needs to be done there as well. And of course, making use of sequence information, right? That's key, I think, to be able to truly get the value out of what that can hold. And then I would also argue the refinement of methodology related to including this into genetic prediction. I've heard some people argue that it's not perfect yet. National cattle evaluation isn't perfect yet, is it? It's an evolving thing. It's not static. It's continually being refined, and that is no reason to stand on the sideline and wait for something to be perfect. The nice thing about breeding and genetics, we know our predictions aren't perfect. That's why we quantify it with accuracy. Okay, so I value your feedback, and if you didn't grab one out there, there's a survey, and I would like you to fill that out. I know Bob asked how many of you were at BIF. We're hosting BIF next summer, about a year from now, in Lincoln, Nebraska, June 18th through the 21st. And with that in mind, Megan Rolfe, host of this year's BIF, kindly sent me the evaluations, okay? And so I perused those because I do want to know what people thought of BIF. When you peruse those, you get the good and the bad, right? So I got into the bad column, and I found this one. Okay? Let me back up. For those of you that don't know what condescending means, I'll give you a chance to ask somebody and look it up. Again, I think the industry's done a tremendous job at technology adoption, particularly those of you in this room that have truly been the first technology adopters in many cases and truly led the charge. To my grad students in the room, you get to pick one, right? So to summarize, in a upbeat and non-condescending tone, phenotypes, and I'm preaching to the choir here, are still critical to collect. And SNP assays are evolving. I think right now the 50K has been the scaffold, if you will, for what we're doing. But scientific community and industry need to work together to make sure that we're able to capitalize on legacy things that have been done whilst moving forward. Many of you represent breeds that have done a tremendous effort in building training populations for traits that are routinely recorded. And it's nice to see so many breeds have also jumped on board and are building those. And the nice thing is, once it starts, right, you can continue building that resource population because we know we're going to have to retrain. Genomic information has been shown to increase the accuracy, particularly for young animals. And it was nice to see the clicker responses. That's where the benefit is, those young, unproven animals. I see representatives from AI studs. Why are they interested in buying animals that have been genotyped? Mitigation of risk, right? Mitigation of risk. Methods will be refined over time, all right? And I'm a perfectionist. John Pollock's a perfectionist. Mark Tholman, Jerry Taylor, Steve Catchman, right? We're all trying to refine this technology. But again, national cattle evaluation is a result of evolution. The difference here is, I would argue, this evolution is happening right out in the open in front of your eyes. And I think that's an exciting thing, that you get to take part in the refinement of this technology. So with that, again, in an upbeat, non-condescending manner,